So let's move on to high heterogeneity. Poor connections can also result in ADCs with variable payloads or a heterogeneous ADC pool that doesn't consistently deliver the same amount of drug, which results in reduced or inconsistent efficacy. So if we could talk a little bit about what the implications of that are and how the pursuit of homogeneity is being addressed in PD. And Monika, perhaps I don't know, maybe you can speak to the adoption of maybe, or application that is of computational or AI tools in that effort. But in terms of the implications and how the pursuit of homogeneity is being addressed in PD, Philipp or Léo, would you like to tackle that one?
I can take it if you want. Yeah. So yeah. I think the main issue with heterogeneity, you can have too low DAR species or too high DAR. I mean, the low DAR are not so potent, but in some cases, having naked antibody or very low DAR species may not be an issue because it could help also tumor penetration. But I think the too high DAR, if those ADC species are too lipophilic, then you can start having very, very high clearance because they will internalize cells unspecifically even without targets, because the lipophilicity of the payload will have a strong affinity for the lipophilic membrane of cells, and that can cause high clearance, high uptake in the liver, high clearance, but also a lot of toxicity, particularly in the liver. So you don't want having ADCs that have too many drugs way beyond your targeted DAR. Yeah? So I think that is very important to control that, and your process should allow you to do that. And with respect to AI, maybe it could, I mean, at least some computational tool could, if you go for a really random conjugation, an old style conjugation, maybe if you use some experimental, computationally aided design, you'll be able to maybe to play a bit on your DAR distribution, but there's just a certain amount of DAR restriction you'll be able to achieve by playing on your conjugation condition, and then you need to adopt a strategy to reach high homogeneity, either saturation of your conjugation site or go for a site-specific conjugation technology. Yeah.
Monika, is there a pathologist perspective on that topic?
I would say that, as Léo mentioned, a computational assessment regarding, de novo design using generative models could be used, or a structure-based design could be used. So that could help with the question which we are having right now. So apart from that, as far as I would say the way the ADC is designed as far as homogeneity is concerned, I think Léo has already mentioned what needs to be done there.
Léo, you talked about, I want to address another audience question from Harch here. Harch has a lot of questions for us. You talked at the outset a little bit about finding a one-stop shop, and Harch is curious about your thoughts on going to specialized antibody, drug linker, conjugation, and fill finish CMOs versus CMOs that cover two or more of the manufacturing steps. So Léo, is that something you'd care to comment on?
I mean, going with those type of partners, I think brings a lot of advantages because your logistic will be much easier, and sometimes you may also avoid redundancies for incoming controls and of the different intermediate you may ship, your linker, your antibodies. So playing with a partner that is a real one-stop shop, that can be helpful. Unfortunately, there are not so many, and sometimes I think they might be as well a bit more expensive for your development. So it also depends on the potential of your product and the speed of development you want to achieve. I think you have to balance everything there.
Yeah. Philipp, is that something you can comment on at Araris? Is there a sort of a philosophy around selection of outsourced partners and trying to consolidate some of this activity in under a single roof?
Yeah, I mean, I can only echo what Léo said. I mean, when you have a one-stop shop, it facilitates a lot of things, because everything comes from one hand. You don't need to deal probably with logistic and managing all of them separately. While when you have everything separately, you need to really it may take a lot more efforts to manage all of those separately, and there may be redundancy as well, what Léo said. On the other hand, of course, if you have different providers, then you may also have more flexibility in exchanging them, so you're not bound by one. So I think it's a business decision whether you want to work with a one-stop shop or with several providers. So and other resources, of course, ultimately as well. So.
Yeah, I'm curious if you survey that outsourced landscape, is there so logistics. Setting logistics aside, and I understand it's a giant consideration, has profound impacts on the business. But if you set logistics aside and you survey the outsourced manufacturing environment, is there a thing? Is best in class a thing? Like, would you ever make the decision to say, no, we are going to actually split this up and do some things with this CMO and other things with a different CMO because they have some best-in-class capability that perhaps the one-stop shop or the 2 or 3 stop shop doesn't have?
Well, so if I may start here, so
yeah,
I mean, from what I hear, I think it's a business decision, and I think probably it depends also on the complexity of your project. So if you have let's say conventional conjugation, like a random conjugation or cysteine conjugation, then which I would say is well established, you may go with a one-stop shop, let's say. And when you have a more complex molecule, when you really need specialized, like let's say you have a very special conjugation that is very unique to you, or exactly, then you may rely on special sort of outsourced capabilities. So I think it depends from case to case what makes sense for the company. So.
Sure.
Yeah.
I completely agree there, Matt, because I think there's always an interplay between, what the present need is, also what turnaround time, the costs involved. So I think these are always an interplay of different factors when making that decision.
And there's also your own historical background. If your linker chemistry is known by a particular CDMO, it will be much easier to redo another batch just at the same place, very quickly. And as Philipp mentioned, if you have a partner for your specific conjugation and they know your process, it's a no-brainer as well to go there. And then you're in that. I think in that context, you should accept to divide a bit your logistic, even if you're in 2 different countries, because I mean, custom can delay things, but sometimes it can be worth it if everything is is robust. It may also avoid you a big mistake or something that will impact your timelines and take 6 or 12 months in your project because you failed something. So
yeah. Geographical considerations also are very, very important, as outlined. I think that also plays a big role in getting your, things on time and how processes might affect the delivery of that. So I think that is also a very important point in considering.
I'm really good at asking questions that the default response is, well, it depends on a lot of things. Miles, let's take a look at polling question number 2. This one again is for the folks tuning in. Speaking of outsourcing, which of the following functions are you handling in-house versus outsourcing? So take a look at those selections. I won't read through them all. You can see them. And make a choice. And I'm interested as we wait for that response, Léo and Philipp, based on your experience, on your experiences, what do you anticipate being handled in-house versus outsourced. Leo, how about you? Do you have any projections on how our audience will respond to this one?
I think manufacturing maybe, because there are companies that are really specialized and handles new ADC project every trimester, so they've seen many compounds, many problems, and they're probably going to be better than your mid-sized company who may just push one ADC project every year or so, So I think if you're a biotech or a mid-sized company, I think in most cases you'll do that, outsourcing manufacturing.
Yeah.
I completely agree. I think when you do manufacturing as a small biotech, you don't want to establish your own manufacturing process in-house. It makes more sense to really rely on experience out externally, of course, because then you can really allocate resources to where you're specialized to, like linker payload design, maybe target selection, or depending on where you're coming from as a company and what your background is, or when you're an antibody. When you have your background in antibody engineering, then of course you focus on antibody engineering. So, but I would say manufacturing process development. But let's see.
Yeah, we'll see. We'll take a look at those results here momentarily. But before we do, I want to get to one related question from John. He's curious about where is the industry on combining downstream purification processing with DL conjugation, as opposed to performing antibody intermediate and ADC production separately. So Philipp, since you're large on my screen, is that one that you can address?
Okay, let me read the question again. So where's the
number 14 there?
DL, I guess DL means drug linker, so conjugation as opposed to from MAB intermediate and ADC production separately. Yeah, I think that as I read it, the question, so I think it depends on your process and your ADC you want to produce. So if you want to use a conventional linker or drug linker like a vedotin, which is, let's say, very well established and well validated linker payload, you may be more flexible in terms of where do you go to a CRO, and many can probably offer that service. In terms of antibody intermediate, I would say it's also a very well-established process to produce the antibody, I would say. If it's a standard IgG, I would say. If it's a bispecific with, let's say, a cross-map, maybe different, because not everybody may have that experience. And if you have a completely novel drug linker, really unique, let's say, release mechanism or polymer attached on it, then you many probably will claim that they can do it, but only few maybe indeed be in the position to actually deliver a very special, let's say, a drug linker. And ADC production separately, again, it depends on whether you have a more standard ADC in mind or a more novel approach to generate the ADC. Exactly. So so it depends on how the ADC is produced and whether how easy the ADC production will eventually be. So based on technology.
Thank you.
I hope that answered the question of John. So,
yeah.
John will let us know if not. Leo, agree, disagree? Anything to add to Philipp's response there?
I think, yeah, maybe the question alludes as well to the fact that maybe you may be able to conjugate your linker payload with a not fully purified antibody intermediates, but maybe using your crude antibody and try to take that shortcut. Maybe this may be possible, using those new cell-free technologies to produce antibodies. There are one or two biotech in the US that do that, and maybe they can produce antibodies with already handles for click chemistry that may give you very clean conjugation chemistry and things like that. So maybe such technologies may allow you to take such production shortcuts. But obviously, you'll be faster. Your process will be simpler, but you may have to pay. You'll be dependent on on someone else's technology if you want to do that. So, or you'll have to develop your own production. But I think it's a field that will be growing in the future. Yeah.
Yeah. Okay, let's take a look at those poll results, Miles. I don't know if we pushed those out yet. There we go. Yep. As suspected, manufacturing process development, 25%. Looks like that's the biggest chunk. And then target selection. Any reflections on those results? So you guys called it, so.
I think target selection may come from the fact that nowadays many companies in license projects, antibodies or ADC projects, so they will rely on a partner that will have done at least the target selection, maybe the lead antibody discovery at least, and so that may explain why we get that type of answer. Yeah, I think biotech are the main innovators in this world, I think, as opposed to very large pharma.
Yeah, we like to hear that. I'm super happy to have 3 fantastic ADC biotech innovators on our panel today.